my exam Module 5 mini video, I chose to do Prompt 5.2, altering the DNA sequence. In looking at the sequence given, we can understand that 4 amino acids are formed from the sequence in vivo. However, before diving into finding the amino acids, it will help us if we first figure out which one is the coding strand, as we must transcribe this into mRNA in order to move forward. To aid us in figuring it out, it helps to flip the bottom strand to identify the starting codon, which we can see as HEG. From this, we can count down the sequence to identify our stop codon. In counting down 3 amino acids, we find TAA, which we know will turn into UAA in our mRNA sequence, which happens to be one of 3 stop codons. Now that we have this identified, we can switch all of our T's with U's in the mRNA sequence to get a better look at our coding strand and identify the amino acids present using the table. Now that we have our coding strand and can convert this into our mRNA sequence, we are able to identify the amino acids present. We can see that AUG is consistent with methionine, GUC is consistent with valine, AGC is consistent with serine, AAG is consistent with lysine, and UAA is a stop codon. In order to create a missense mutation, we must alter a spot in the coding strand and the mRNA sequence that results in a different amino acid, not the same. To demonstrate this, in this DNA sequence, I changed the cytosine in AGC to adenine, changing it to AGA. This changes our amino acid from serine to arginine. This is also a transversion. A nonsense mutation results in a change in the mRNA sequence to make a stop codon. In order to demonstrate this in the sequence provided, I changed the first adenine in AGG to a uracil, causing it to be UAG, which results in one of the 3 stop codons, therefore making it a nonsense mutation. And because we are changing it from A to U, it is a transversion. In order to create a silent mutation, we must alter a spot in the coding strand that matches an alteration of the mRNA sequence so that it ends up with the same amino acid after the alteration. For my example, I chose to change cytosine to adenine, giving us GUA rather than GUC. However, in looking at the table, both of these options work to give us the amino acid valine. It is also important to note that this mutation is a transversion. In order to create a read-through mutation, we must make an alteration in the stop codon to convert it into an amino acid. In order to demonstrate this, I have changed a second adenine in TAA, or UAA in the mRNA sequence, into cytosine, which converts it from a stop codon into the amino acid tyrosine. This is considered a transversion. Additionally, because the stop codon is now gone due to the change, the sequence would not stop and more amino acids would continue to form along the sequence.